Welcome everyone to today's Bite-Size Talk. I'm very happy that today with us is Kübra Nace from the German Human Genome Archive and DKFZ Heidelberg. And she is going to talk about NF-core variant benchmarking. So this is your stage, Kübra.
Thank you very much for the introduction. And let me share my screen. Okay, so all good, I guess.
Yes, we can see it.
Yeah. I am Kübra from GHGA, what we call. I know it is hard to pronounce. And yeah, I'm working as bioinformatician and scientific workflows engineer in the workflows work stream in the team. And today I'm going to talk about variant benchmarking pipeline I created under NF-core. And basically, it is going to be with technical details mostly, I would say. But I also want to talk a little bit about my motivation and what it is, what is the meaning of benchmarking and variant benchmarking. So workflow managers came along our life and they made the reproducibility possible. And that was great for our lives. It was making the scientific world easier, to reperform the analysis. But the accuracy is another issue. And using those pipelines are not mean that the results you have is accurate enough for downstream analysis. So there should be biological accuracy behind your analysis. And benchmarking is what it is all about. And if you need to see the performance of your tool, basically you have to benchmark, which means that you need to compare it with some ground truth and also with the other pipelines, other tools, other experiments, let's say. And benchmarking is about comparison. And there are 4 core elements with benchmarking, which like the first element is truth data, meaning that it is the ground element, like what you are comparing your results to. Nowadays, there are some truth files established. They are very well studied. They are very well experimented and served into the community to use as a base alignment. And the few examples are Genome in a Bottle study. They have published many, like 8 or 9 samples as a ground truth, and HE002 is like one of the most studied ones. They have structural variants for small variants and etc. For somatic analysis, there are also a few samples now, especially from 62 study. There are also others that you can use as ground truth. And there are some methods. Method is the tool you use to compare your result with the ground truth. And this is basically the comparison engine. It is the algorithm itself. And there are also performance matrices you can use as a sign of your performance, like statistical measurements, the number of true positives, false negatives, and some calculations along with them, like precision, recall, and F1. And the fourth core element is how to show the results, how you are going to represent in an understandable way with some kind of visualizations. So these 4 elements I wanted to include into my pipeline. So this task seems to be easy, right? You need to compare your results with the ground truth. And sorry for forgetting, but from now on, I will be talking only about variant benchmarking. There are also different kinds of benchmarking to compare the, let's say, some softwares. You can also compare memory, CPU usage, etc. But this pipeline is only about variant benchmarking. And when we say variant benchmarking, it is really comparing and matching the variants. So if you consider a simpler case with the single nucleated variant changes, the task is just to match the positions and also references and alterations, the changes. Then you can basically find the match. But what about for the more complicated analysis? Let's say you have structural variants, some INDELs, and do you want to just compare with them? But how to make the decision, right? Sometimes the positions might not change, but it may be indication of the same change in the genome. So with this kind of tasks to match, there should be a way of standardization of the rules and sometimes making your own rules. You can say that I am going to match to variants in a sense that they can be nearby, like two nucleated changes and etc. So this will be your own rules, and you will be deciding the number of matches with those kind of rules. So that is what benchmarking tools are doing actually. And there is a problem with basically structural variants, and it is the ambiguity in the allelic representations. So as you can see, so in this presentation 1, 2, and 3, it is all the same change, but different tools are referring them as different changes with different positions, different references, and alterations. So if you need to try to match those, they won't, of course, because these positions are different. What you need to see is to infer to the reference genome itself and try to normalize those samples in a way that they look similar, and then you may be able to just match them. So this is the way of standardization and normalization of those variants. And this is a basic requirement for a good benchmarking. So before I start to do this pipeline, I had actually two motivations. One was coming from the GHGA itself. In GHGA, we are trying to provide a standardized way of analysis for the data we store. So GHGA is an archive actually for Germany. We will be storing the data, like many types of data, and then we will be providing a unified way of analysis for them, which is going to be through some pipelines. And these pipelines, there should be a decision step, like which tool is the best for us. We have to compare the tools itself according to the some ground truths. And then this decision step is important because we want to serve the best for the community. And to do that, we kind of built upon a circle for us to decide which tool to use. And for that, we had to have a benchmarking strategy. And that is why I came up with this pipeline. So in our daily life, we will be comparing the results from our analysis with each other and to the truth set to see if our performance is good enough. And then we will be sharing the configs, the parameter sets that we use for the analysis with the community so that everything is being transparent. And my second motivation was coming from the NF core itself. I saw that like there are many variant calling pipelines in INF core, but the validation was missing for accuracy, but for biological accuracy. And I thought like in INF core there should be kind of a workflow to serve as an accuracy downstream process serving to those pipelines. And that was actually my two motivations to develop this pipeline. And yeah, this is the kind of summary what pipeline does. And it is mainly the tool to evaluate and validate the accuracy of variant calling methods in genomic research. And I mainly used Genome bottle and 62 samples to adapt it as a truth golden set. But actually, you can compare any two variants if you want to use this pipeline. It provides standardization and normalization tools in order to remove this ambiguity in variant representations, but also it tries to reformat the VCF files. Sometimes different variant callers tend to report the variants in different formats, and they sometimes miss some formattings, and my aim was to be able to fill this gap as much as possible. And it can be used for small variant benchmarking like SNVs, INDELs, but also for structural variants and copy number variations. I have different methods, algorithms for comparison benchmarking strategy, and those are includes HAPI, RTG tools, SOMPy, SV Analyzer, TrueVary, VTier, and what I called as Intersect, which is actually BCF tools. And it is able to use both germline and for somatic calling VCS. Now I will show you how to use it. Basically, the main requirement for the pipeline is to provide a truth VCF. It came along with the truth ID, which is going to be used as the sample name for truth truth as the truth header. One can use a regions bed or targets bed, and this differentiation is based on BCF tools. BCF tools has two regions, like one with minus R, one with minus T, and that is the differentiation. And the sample sheet is includes the test VCFs together with the ID, VCF file, and name of the caller used to call the variance itself. So this is the main requirement. And this test VCS can also be multi-sample. Example is here. So if this test one VCF involves more than one sample, the only need is to describe it in the subsample column with the name, and then it will subsample and use it. The references, we have a column parameter for stratifications. So in benchmarking, it is common to use stratification files to restrict the analysis to certain areas, like difficult to call regions, low complexity regions, or repetitive regions. It really depends on the type of analysis you attempt. And it is possible by this, and it came along with FASTA file. And the mandatory parameters are this. So one has to define the genome to use, otherwise the FASTA file will be required. The analysis type can be either germline or somatic, And I only allow for one analysis per variant type. So it has to be either like structural, small, or copy number. And there can be more than one method. One can select all the methods can be used for structural variant analysis. And then it will came along with truth ID and truth VCF. Yeah, this I already explained. And next, uh, the next option is with liftover, but liftover is not mandatory. It is only needed if one or more files, either if it is true or if it is in test file, is not in the same genome build. It is something common actually to have different genome builds when you need to do comparisons. That's why I have this option to lift the VCF over, and it is automatic. If the liftover is truth, then the truth file will be lifted over. And if it is in the test, there is this column in the sample sheet with liftover. If it is true, it will lift over this file to came along with the same build and will perform the analysis. Next step is variant standardization. This is only for structural variants because they have more representation issues, basically. Now I have two options with SYNC and homogenize. These tools are like they are ready tools. I'm just using them to standardize the way of representation. For example, to normalize um, insertion. For example, sometimes the duplications are being named as insertion, but sometimes duplication. My main aim is to standardize the way to name them and also the formatting issues here. But it is all optional and it won't be applied unless the user chooses to apply so. And I have some plans to involve more tools here, but it's on my to-do list. And the second step is also optional for variant pre-processing. This can be applied to both small variants and structural variants. It actually uses many BCF tools sequentially. One is to split the multi-allelic variants. So there are some different strategies to use multi-allelics in the benchmarking. One can choose to use them together or in split version. And there is also option to deduplicate those, like which one of them to keep, one of them to delete. And one can choose to use BCF norm option to left or right align the variants. And PrePy is basically the tool supplied by Illumina, and it can be also applied. And there are also options to completely split SNV and INDEL variants here. The next option is about filtering. So filtering is actually among normalization. I personally wanted to refer filtering as a step in the pipeline itself because you have to apply the same filtering to all the samples together if you want to be consistent with the results. And this is basically applying SV survivor filter for structure variants and BCF dose filter for other variants, for small variants as well. And the only option is to use exclude or include. This is coming from BCF dose. One can use any expression and apply the filtering here. And like some examples could be to filter out difficult to call regions here, or you can basically apply a filtering strategy so that you get rid of the low mapping quality variance, and like anything can be applied actually here. And the last step before benchmarking is gathering the variance statistics. I think this is important because you need to know how many variants you are using to benchmark. And this is using BCF tools for small variants and Survivor for structural variants. Next it comes benchmarking. So as I mentioned, I'm using different methods for different variant types. And sometimes for germline and somatic calling, it also differs. For germline variant calling for small variants, I have HAPI and RTG tools. And for somatic variant calling, instead of HAPI, there is a modified version of HAPI called as SOMPI, and I'm providing SOMPI. The only difference is with the report, actually. SOMPI takes into allele frequencies account in the reporting so that you know which allele frequency bins you have as separate number of true positives, false positives, and etc. And for structural variant benchmarking, actually, I don't have different tools for somatic analysis right now because basically there is no readily available tool reporting allele frequencies. But I have plans to stratify those in later steps, probably in the downstream analysis. And now I have TrueVary, SV Analyzer, and VTIR for structural variant benchmarking. And those all have really different strategies to make the benchmarking. They have different parameters inferring to different types of comparisons. I think this is really important for this pipeline, like making it flexible to apply different algorithms. One can use all to see the differences, and I am testing this pipeline for a long time. There are differences. It's mainly because of algorithmic changes, but yeah, that's it. And sometimes, so here, TrueVary, I think, is more popular than the other two tools, and it is only because TrueVary is more flexible and can be used for uh, structure variants without uh, the sequence itself, like ambiguous variants, I would say. And yeah, came along with PCT-SEC. So I implemented the benchmarking tool parameters to the sample sheet itself, and there is a reason behind it. So those parameters can be used differently per test. So those can be tuned well into the testing strategy. So if you know your testing sample very well, you might need to apply this parameters. For example, in PCT-SEC, when it is 0, it means that do not consider the sequence itself, but just do the comparison amplified from the genomic region itself. So this may give you an idea. Of course, it is not going to be accurate, but it is going to be a result. It is the same is applicable to SV Analyzer as well. I also put the parameters to sample sheet, and it can be changed by tool. The other tool is interesting here is VTIR, because VTIR is the only tool can be used for copy number variation analysis. VTIR in the VTIR report, all the different SV types are separated, and it provides a good table with it. The only difficulty is the files has to be well formatted accordingly, and it's sometimes tricky to make it used. Yeah, that's it. And I so it was only for copy number variations actually. I was searching for benchmarking strategies what to do because like copy number variations are not always coming together like inside VCF format, but they are usually in a bed formatted files. They provide the regions and then indicate the number of copy numbers per region and sometimes the other information as well, but that's basically it. And if you want to benchmark it, according to the research I did, like with the other papers, they just try to intersect the regions. And this is mainly together with BCF tools. That is why I provide this branch to make the intersection analysis per region. And this is an example. For example, in here, get the K-man results in a CSV format. And the intersection tool that I implemented basically tries to intersect with the target region and then calculates the performance matrices accordingly. And finally, we have downstream analysis tools. This is basically all about merging the results and trying to make a good visualizations ahead of it. And I will try to summarize it as a little bit. And this is the output directory structuring. The main, so the results are coming along with variant type, let's say small, and you have the benchmarking, pre-processing, and statistics results per sample. So you have the initial variance statistics, if it is BCF tools on Survivor. You have pre-processed VCFs here. So this is the VCF file used for benchmarking at the latest steps. And finally, you have all the benchmarking results per tool. It will depend on which tool you use, but it is going to be all listed here. So you all have the raw results basically. And this is for sample 1, sample 2, how many samples you have, you will have those results. And you also have it for truths, but benchmarking is not, of course. The pre-processing step will be give you a chance to compare the initial variant numbers. And the actual summary, I have a step to make the comparisons between true positive, false positive, and false negative variants. This is quite useful because like in here, as you can see, there are 3 samples and those are all, I guess, from true positives. I'm not sure, but yeah, in here you will be actually seeing the comparative results. For example, if you're interested in the true positives only called with your own tool, this is the file you can observe those. And if this is only possible if the benchmarking tool results with separate true positive, false positive, and false negative files, but it's on my list to provide for all strategies. And this is basically the table out of it. It's just merging the results per file and giving you the TXT formatted file with tool, the number of true positives, false positives, precision recall, and etc. Whatever it is available in the benchmarking result, I would say. And then we have plots. Now plots are not great, I would say, because I didn't have time to just make it better. But the general idea is to provide a plot to visualize the true positive, false positive, and false negative numbers accordingly with the precision recall and F1 matrices. But this is actually better because we have data wizard tool in the report. It is an HTML file. It is interactive. So when you open the file, you actually have these all columns, and you can use filtrations and generate your own tables. It's not embedded here, but it is also possible to add plots directly to data wizard. So it's going to be a good addition. And I have some like test configs. I also embedded NF tests for pipeline testing. And if you ever run and encounter an error and bug, just let me know and please let it let it run. And yeah, those are my feature improvement strategies, let's say. Right now, I don't have quite well somatic variant benchmarking for structure variants, which I intend to do. And I wanna make it with better visualizations. Family trios are all on my to-do list, but to be honest, I'm not sure if it is something needed, required from the community. And I am thinking about adding coverage stratifications, some new tools for standardizations, maybe for benchmarking. And now I'm in the process of writing a small white paper along with it. Yeah, this is the workflow workstream team. And I would like to thank especially to Nicolas because he's like all the way along with the revisions with this pipeline. And Maxim is also helping me a lot. And lately, Kreshimir was kind enough to fix my logo. And yeah, my teammates Florian and Samish, and thanks to the all NF Core community to make this pipeline possible. Yeah, that's will be all. Thank you.
Thank you very much. That looks like a lot of work. So anyone who has a question can now turn on their video and unmute themselves. So if there are any questions, please ask them now. Maybe I'll make a start. I was always wondering, how do you know that your ground truth is actually your ground truth? Like how this could change, right, with new analysis methods or so on. So when do you know you have to move on to a new ground truth?
I think it is a decision to make yourself, I would say. I mean, the new ground tools are, I would say, usually better. There might be like multiple reasons behind it, but probably the new versions will be better. But I guess it also depends on the formattings with the files, because like with formats in VCF files is always an issue, I would say. Yeah, but there is no way to know, to be honest. You just need to compare it, I guess, to see the result.
Okay. So you just decide on something to be the ground truth, whatever it is, and then compare everything to that.
No, I mean, it has to be coming from the same sample. Like
Yeah.
if you are planning to do an HE002 analysis, your truth has to be the same. So basically, you need to be analyzing the same set of samples, but like which versioning, I wouldn't know. You just need to be in parallel.
Do we have any more questions from the audience?
Hello, I guess I have one.
Yep.
Yeah. So it follows up to your what you mentioned regarding ground truth. So, for example, we normally look at human genetic data, and we always say that, yeah, humans are diploids, so we allow two different zygosities.
Mm.
But in the case in cancer, for example, where you have a cancer sample, and the cancer sample is made out of a lot of different cells. And
Yes.
so do you allow for like maybe three or even all four possible nucleotides there as the ground truth?
Yeah, somatic benchmarking is really difficult, I would say, because there are not too much ground truths along with it. Right now we have sexy too, but it's not that as detailed, I imagine. It's both about ground truth and also with reporting, right? I mean, your comparison strategy has to be flexible enough to catch those low frequency samples in the first step. And second step is to be able to report it with the correct matrices. And the tools right now are not working accordingly. I mean, you have to implement your own strategy, I would say. But yes, this is something of missing in this pipeline. My aim is to be able to go along with this, but it is really, I would say, young research now.
Yeah, thanks. Yeah, because I'm also currently working on something similar where I am taking the results of a lot of different variant callers. And yeah, then the question is, do I filter them for like two different zygosities, or do I just keep everything? And I think that is where your pipeline is going to be helpful
Yeah,
for me.
maybe you can try to make like you can split the multi-allelic regions, but not deduplicate it and try to match it all. But you have to be careful with your matches because if it is really flexible, they will match both, and you will basically will duplicate the true positives.
See.
Yeah.
Yeah, that's a good point. Thanks.
Okay.
There is another question from Fabio in the chat. He thanks you for the talk, and he has out of curiosity, he asks if you considered also can oncoliner.
Oncoline, of course, I did. Yes. I was planning to edit, to be honest, but I had some issues and errors with multiple reasons, with images, containers, and then it was yeah, it's still on my to-do list, but yeah, I have to resolve those issues first. But yeah, I know the tool.
I hope this answers your question, Fabio. Are there any more questions from the audience? If not, then right, he thanks you. Then I would like to thank you, Kypra, specifically for giving this talk.
Thank you.
It was very interesting to hear about this new pipeline. And I would also like to thank everyone who was in the audience for listening in today. So thank you very much, and I hope to see you for our next bite-sized talk. Bye-bye.